Good evening, everyone, and welcome to the third of this year's Oxford Botanic Garden and Arboretum Autumn Science Lectures, sponsored by Plants, People, Planet and the New Phytologist Foundation. The theme of this year's lectures, as those of you who've attended the first two will know, is paleobotany and the early evolution of land plants. In case you don't know me already, my name is Simon Hiscock. I'm the director of the Oxford Botanic Garden and Arboretum, and as usual, it will be my pleasure to introduce tonight's speaker, but only after some preliminary housekeeping information. Firstly, this lecture is being recorded, but your camera and mic are turned off, so you are not being recorded. If you would like to ask a question to the speaker, please use the Q&A function at any time, and I will ask your question at the end of the talk. If you need to contact us about anything else, for instance, technical issues, then use the chat function and one of the team will get back to you very quickly. The recording will be made available for all attendees after the event and it will come to you via the email that you registered with. So now it's my great pleasure to introduce tonight's speaker, Dr. James Clark, Prize Fellow in Theoretical and Empirical Evolution at the Milner Centre for Evolution at the University of Bath. James's research spans the fields of macro and microevolution, from paleobotany and phylogenetics to the genes and gene networks involved in phenotypic plasticity and adaptation. A common thread in all of James's research is the use of state-of-the-art genome analysis. James studied as an undergraduate at the University of Bristol before taking a master's in ecology, evolution, systematics and population biology at Imperial College. Then he returned to Bristol for his PhD. He then did a postdoc with me here at Oxford, studying gene networks involved in phenotypic plasticity in Mount Etna ragworts. He then returned to Bristol for a postdoc studying the evolution of stomata, and in 2023 he moved to the University of Bath to take up a prestigious prize fellowship that has allowed him to establish his own independent research on diverse aspects of plant evolution. In his talk this evening, James will discuss how insights from plant genomics, new scientific approaches, and the earliest plant fossils are shaping our understanding of the evolution of the first land plants. Welcome, James, and over to you. We're very pleased to have you here this evening.
Wonderful. Thank you very much, Simon. I'm just going to share my screen. So that should now be visible. So hopefully it is, and I'll just dive straight in. So the talk that I'm presenting to you today is all about the tree of life, the relationships among different species, this field called phylogenetics, and how this can guide or influence our view of the origins and the early evolution of plants. So I'm going to start off with hopefully quite a light touch introduction to phylogeny, but perhaps a necessary one, before talking about the algal origins of land plants, the relationships among plants themselves, and then to try and tackle this kind of big question about the evolution of the very earliest plants. So, phylogeny is first and foremost the study of the relationships among organisms. And one of the kind of more famous kind of depictions of phylogeny is this one here from Darwin himself, showing how the relationships among species can be shown through these branching diagrams or phylogenetic trees. So the branching diagram is not just a schematic or a representation. It also communicates something of our view of the evolutionary process. So in this simple kind of diagram, as a lineage is existing over time, we know that sometimes new species will form through the process of speciation, creating a fork in the path of a lineage as it splits into two and begins to diverge. And as more and more of these splits occur as time goes on, it starts to give us the basis of that tree-like shape with branches connecting different species and leading to these tips. And the series of branches are kind of more often illustrated like this as a phylogeny, and it depicts the order in which these species have diverged and in turn how closely related different species are to one another. So in this case, the most recent branching gave rise to species C and D, which are the more kind of the most closely related or sister lineages. And these are then more closely related to species B than they are to species A. And also within our phylogeny, our phylogenetic tree, the points where the two branches meet within the tree, these represent the common ancestors. So this first circle being the common ancestor of C and D, and then B, C, and D, and A, B, C, and D. And this is just to show throughout the talk as phylogeneticists, we have a kind of a bit of a bad habit of showing trees in various forms, often without reason. So sometimes they'll branch upwards like they did in the previous slide. Sometimes they'll be branching downwards and sometimes sideways. But fundamentally, they're all depicting the same thing. And in fact, if you were to kind of brave a trip down the A420 to Bristol, you would even get to see a phylogenetic tree in real life with the angiosperm or flowering plant phylogeny display at the Botanic Gardens, which of course was the first of its kind developed under Simon's stewardship. So the field of inferring species relationships, inferring phylogenies is phylogenetics. And I think there are just kind of a few fundamentals that I think would be useful to kind of talk about before I kind of dive into the plant evolution. So the first thing is to note that we tend to rely on kind of two main sources of information to work out how species are related. So this is kind of the morphology or the traits that are present in different species, or the underlying molecular sequence data, be it kind of DNA or amino acids. There are also a few kind of important principles that will come up throughout the talk. And the first of these really is just a way of grouping or dividing species within phylogenies. And the first of these is a concept called monophyly. So monophyly describes a set of species that includes its common ancestor and all of the descendants. So in this particular example, we can start with the common ancestor and we can find a clade that includes A, B, C, and D, forming a single monophyletic group. So a nice example of a monophyletic group would be primates, since it includes here the lemurs, the chimpanzees, and the humans, and also their common ancestor. But an alternative to monophyly is paraphyly, and this is the state where we include the common ancestor, but not necessarily all of its descendants. So here, if we were to devise a group that included A, B, and C, but not D, then this would form a paraphyletic lineage. So a really good example of paraphily that we tend to see a lot is the concept of reptiles. So reptiles are kind of commonly defined as the lepidosaurs, so the lizards and the snakes, the turtles and the crocodiles, but we tend not to count birds as being reptiles. So the concept of a reptile is paraphyletic. There's also a slightly more unusual kind of phylogenetic pattern, which is polyphyly, which includes a group which doesn't actually include the common ancestor nor all of its descendants. So it's kind of a bit of an evolutionary mess. And the best example of this I can find is the field of herpetology. So this is the study of amphibians and reptiles, themselves paraphyletic. So while herpetology is certainly worthwhile, it is a polyphyletic field. So then I'll very briefly talk a little bit about how we construct a phylogeny. So the first source of information, morphology, often requires us to identify the shared presence or absence of different traits among species. So in this case, we have three species, an oak, quercus, a ginkgo tree, and sphagnum, a peat moss. And the shared presence of lignin, woodiness, and leaves in the two tree species tell us that they may be more closely related to one another than they are to the peat moss. And then likewise with molecular data, rather than the kind of physical traits or attributes of a species, we look at variation in the molecular alphabet of the DNA or amino acids that are present in their genes to try and identify common substitutions or changes. So again here, with our oak, ginkgo, and peat moss, we can see that there are two kind of shared substitutions at two different sites in the sequence that might suggest that the oak and ginkgo are more closely related to one another than they are to the peat moss. So as a final kind of point of introduction to phylogenetics, what I'd really like to impress on you in this talk is the idea of tree thinking, or how phylogenetic trees can impact our view of evolution. And maybe a fairly unlikely example of this, but we're fairly confident that humans and chimpanzees are closely related, at least more so than they are to lemurs. But if you were to imagine a brand new study, perhaps, that proposed the opposite, that humans and lemurs share a more recent common ancestor than they do with chimpanzees, then what this would mean for how we understand human evolution. So how would we then understand the evolution of human nature or our behavior or our diet, given that we might be more closely related to a lemur than we are to a chimpanzee? So with that kind of hypothetical scenario, I'm finally going to get on to plants and to specifically consider what a plant is. So not necessarily in terms of the diversity of plants, but more from an evolutionary perspective. So plants are first and foremost eukaryotes. So eukaryotes are kind of one of the major domains of life, and this is the diversity of eukaryotes. So as animals, we sit within the Opistheconta, but plants sit over here in the Archaeplastida. So the Archaeplastida are more or less unified by the presence of chloroplasts. So a secondary endosymbiosis event during which a kind of an ancient cell entrained cyanobacteria to turn it into this kind of fundamental sugar-producing structure that typifies all plants and algae. And within the Archaeplastida, we find the origin of plants. But here I mean plants, sorry, within the broadest sense, encapsulating both the kind of large macroscopic land plants, but also their algal relatives, the chlorophyte algae. So this is Chlamydomonas. It's a fairly typical green alga. And perhaps the most apparent difference between Chlamydomonas and a land plant is that it is unicellular. So the origin of plants is typically seen as kind of one of the major multicellular evolutionary events in the history of life, with parallels to what we also know has occurred in fungi and in animals, with the origin of complex macroscopic forms from relatively simple unicellular relatives. And this gives us the basis, I suppose, for what is the narrative of plant evolution. So, from a unicellular alga, plants have evolved increasing complexity through to the present day, while moving from marine through to freshwater and eventually onto land. Um, and it's actually kind of here during this freshwater phase that we start to have a bit of a closer look at the origin of plants. So within the green algae, there is a fundamental split. On the kind of left, we have the chlorophyte algae. These are your kind of unicellular things such as Chlamydomonas, but they form a sister group to another type of green algae called the streptophytes. And it's here that we find not only plants, but also a bunch of other algae. And at first, certainly for me, when I look at other streptophyte algae, I kind of struggle to see the difference between these and the chlorophyte algae like Chlamydomonas, especially among some of these kind of unicellular examples on the top. But you can also start to see a bit more diversity in their growth forms, from some of these kind of filamentous kind of chains to some of these more intriguing macroscopic forms as well. And it's from this diversity of different growth forms, we can perhaps begin to piece together a scenario for the assembly of the land plant body plan. So maybe from a simple unicellular form, such as mesostigma, through to the evolution of these filamentous chains like Desmidium here, then later to species that are able to grow as kind of multicellular discs or mats like Coleochaete, and ultimately to the kind of more impressive algal species such as Chara, which is capable of kind of apical, upright, three-dimensional growth and branching, much like we see in land plants. And this is also what the phylogenetic analyses were telling us. So these phylogenies produced kind of around the late 20th century tended to be based either on morphological or biochemical characteristics, reinforced this view of a simple unicellular ancestor within the streptophyte lineage that progressively became more and more complex through to the origin of land plants. And you can even pinpoint some of the major innovations as they proceed along this phylogeny. So from the origins of kind of more simple forms of multicellularity to some of the characteristic features of plants, some of the kind of cellular features that define plants here, and even through to the origin of kind of complex 3D growth, branching, and apical forms that unite things like Chara and land plants. But there was a bit of an issue in the way that some of these phylogenies were put together. And this really comes down to some of the limitations of trying to infer species relationships just using morphology. So the first is the number of characteristics or traits that we can observe. So hopefully more than three, but for unicellular algae, it certainly becomes difficult to identify enough features that we can use to pull apart these different evolutionary relationships, especially given that some of these lineages may have been evolving for over 1 billion years. Secondly, a bit of a kind of methodological concern that I won't go into too much detail, but it turns out that from a computational perspective, it's quite difficult to come up with a realistic model of morphological evolution. And finally, we have this kind of issue of homology. So homology just describes our ability to determine whether traits or features of different species are equivalent among different species. So in this example, the forearms of humans are equivalent to the front legs of a lizard or the wing of a bird or perhaps the fin of a whale. but we know that they're fundamentally different to the arms that we see in an octopus or a starfish or an insect. In plants, we kind of see lots of these different kind of difficult situations to do with homology. So for instance, while we might be confident that the leaves of an oak and a ginkgo tree are equivalent, it's thought that the leaves of ferns, such as this spleenwort here, may have evolved independently. And so independent and convergent evolution of traits can be quite misleading and certainly very difficult to identify across lots of different species. So instead, we tend to look more towards molecular sequences. So here we no longer have this issue of homology. We're a bit more confident about which genes are which and about which parts of this DNA alphabet are equivalent to one another. We can also apply much more complex models to analyze molecular evolution, since we have a better idea of how DNA evolves than we do morphology. And finally, unlike morphology, which perhaps has kind of a finite number of elements that we can look at, eukaryotic genomes contain tens of thousands of genes, which kind of provides almost a limitless number of information for us to analyze. And so even before the advent of genome sequencing, before the development of complex models of molecular evolution, when analyses were restricted to perhaps a handful of genes and relatively simple models, we found something quite strange. And this was that rather than the complex multicellular lineages such as Cara, the analyses were pointing to a different group as being sister to the land plants, and that is the Zygnomatophyceae. So the Zygnomatophyceae, to be honest, are kind of mostly floating pond scum. They're morphologically really simple, most of them being unicellular, while a handful of them have gone on to evolve these kind of simple filament forms. Meanwhile, the kind of previously inferred sister to the land plants, the Carophycean algae with this kind of complex 3D growth, were actually found to only be a distant relative. And this result has actually withstood kind of multiple analyses from different research groups, different data sets, models, sampling of species. And consistently, we find that yes, kind of the earliest branches within the stratophytes are these kind of more simple unicellular species, but the closest living relatives of land plants is more simple and not complex. And so without wishing to kind of overstate kind of what a big deal this was in plant terms, this is maybe equivalent to this kind of lemur chimpanzee reversal situation that we might imagine in primates. And it kind of forces us to reconsider the whole evolution of traits and what we think of as being kind of stepping stones to building a plant, and ask whether they now must have either evolved kind of far earlier than we thought, or have they perhaps evolved multiple times? And if so, how likely is this? And I'd also like to kind of highlight kind of a couple of more recent developments that emphasize just how strange the evolutionary pathway to land plants has been. So first of all, we have a very recent study looking at one of these particularly strange macroscopic lineages, the Coleochaetophyceae, which here shows this kind of 3D disc-forming morphology that was previously thought to be kind of one of the kind of main steps on the way to complex life on land. And this study kind of looked in much closer detail at this family. They looked at how different growth forms had evolved, and they found that actually within this family, if you sample more broadly, they found that the earliest members of this lineage actually had a much more simple, almost unicellular life cycle that then seems to go through these kind of familiar steps of then becoming colonial or filamentous before eventually forming these kind of more complex morphologies, but much more recently than was previously imagined, suggesting that what was previously thought to be kind of an ancestral example of kind of complex growth is actually something that has evolved much more recently and independently only within this lineage of algae. The next example kind of concerns the specific process of the move to land. So one thing we know is that the move to land itself for plants was very stressful. So it involved having to evolve a set of innovations that could allow plants to survive a much more stressful environment. So with elevated UV, no longer kind of abundant water. So people have hypothesized that kind of associated with this algal to plant transition, we should find the molecular machinery required to survive on land. So lots of UV defense, lots of stress response pathways. And now that we have access to the genomes of algae, we can start to ask, when did these stress response pathways evolve? And what was quite surprising, and I would definitely recommend ignoring all the details on this slide, but this is showing kind of the assembly of various stress response pathways within the genomes of algae and plants. They found that actually a lot of the molecular machinery required to respond to the stresses associated with the move to land were present in the algal ancestors of plants, evolving far more early than we had ever previously thought. So together, these findings suggest that while the origins of plants and the process of terrestrialization was a unique and singular event, plants have only evolved once, it happened from within this clade, the stratified algae, where multicellularity and terrestrialization seem to be happening much more frequently than we first thought, suggesting that kind of somewhere within this clade there was almost an evolutionary priming for complex and terrestrial life. So now, hopefully, we have a bit of a better idea of where plants have come from. And like I say, we still believe the origin of plants was a kind of a singular, unique evolutionary event. But the pathway to plant evolution is far more convoluted than we first thought. So we're now going to look at land plants themselves. So as I said before, we have this notion that around the time that plants were diverging from their algal ancestors, the process of terrestrialization, they were moving to land, they acquired a whole suite of new innovations. So these include things like a multicellular sporophyte, so a complex diploid generation, embryogenesis, which kind of gives them their kind of technical name, embryophytes, a thick waxy cuticle, which kind of coats their cells to stop them drying out, and a bunch of other kind of morphological, biochemical, and cellular features, which help them to move onto land and reduce the loss of water. And at this point, I suppose plant evolution could really be boiled down to a series of different characteristics that have helped plants to tolerate increasingly stressful conditions and become less and less dependent on water. And we can visualize kind of the appearance of these traits along our phylogeny, starting with this kind of burst of innovations associated with land, then the evolution of stomata, tiny valves that help plants regulate water loss, increasing complexity of this kind of more and more free-living diploid generation, and then all of the kind of classic plant traits like leaves, roots, seeds, and ultimately flowers. And this kind of leads us to kind of the bulk of plant diversity, the vascular plants, which kind of really make up both the majority of plant diversity and also what we typically think of as being plant-like, with the features that I'd say most people probably associate with plants. So kind of broad leaves, flowers, seeds, roots, and things like that. And this kind of view of plant evolution was very much prevailing for a long time, but it was also centered around a particular phylogenetic hypothesis or framework, one in which a series or a set of plants, the non-vascular plants, the mosses, the liverworts, and the hornworts, formed a paraphyletic grade. So here we have common ancestor of plants, but not including all of the descendant lineages. And within this kind of paraphyletic grade, we have the successive divergence of these different non-vascular plants, the liverworts, mosses, and hornworts, before getting to the vascular plants themselves. And this specific arrangement is really important because it really shapes how we view early plant evolution. So if we were to ask a big question such as, what was the nature of the earliest plant, then any attempt to kind of predict or reconstruct that plant's form or features would have to pass through these successive grades of non-vascular plants. So starting from the vascular plants, you would work backwards and backwards, kind of being more and more informed by these non-vascular plants. And this is even more significant because non-vascular plants share a typical set of features or the absence of them. So they lack true vasculature. They tend to be very small. They have kind of a relatively simple sporophyte, so the gametophyte. The haploid generation is dominant. They tend not to branch, and many of them lack these important air release valves, stomata. And we also know from their genomes that they tend to possess fewer genes than do the vascular plants. So this would then lead to a reconstruction of the earliest plant that was then, like non-vascular plants, would lack vasculature. It wouldn't have branching. It would have a predominantly haploid life history and ultimately have fewer genes than living vascular plants do. And from this ancestor, we then have a nice and clear narrative of land plant evolution. So across these various non-vascular plants, kind of going towards our more familiar vascular plants, we see an overall increase in morphological complexity. We see the gains of these traits such as leaves, roots, seeds, and flowers. We see a net shift towards this diploid dominant life cycle, the sporophyte, becoming increasingly large and independent. And underlining all of this is an increase in genomic complexity. So one potential issue with this scenario is that the earliest complete plant fossils we have don't really match that expectation. So there are kind of several species shown here, but typically our earliest complete plant fossils are often preserved with both the sporophyte and the gametophyte, so both generations. And most of them have vascular tissue, or at least some rudimentary version of it. They all seem to branch. They all seem to possess these tiny valves, stomata. And what's more is that unlike either vascular or non-vascular plants, these two generations, the gametophyte and the sporophyte, are roughly equivalent. So they're called isomorphic. So really one problem is that the fossil record doesn't necessarily match our prediction of the earliest plants in this scenario. So another potential issue is that 5 or 6 years ago, there was a bit of a revolution in plant phylogenetics. And this was brought about by an initiative called the 1000 Plant Transcriptome Project, or the 1KP. And this project pretty much involved sequencing the transcriptome, which is the whole set of genes of 1,000 plants across various different kind of plant and algal lineages. And this ushered in something which is kind of called phylogenomics, so phylogenetics, but applied at the scale of whole genomes. And the bottom line of this is it gave us something that we had never had before. So lots of genes to work with across lots of different species. And at the same time, new models of molecular evolution were being developed for us to apply to this data. And really all this means, so with more genes, we can increase the signal in our phylogenetic analyses. With more species, we're able to reduce the noise. And with these new models of molecular evolution, we can overcome a lot of the biases that are present in molecular sequences. And so armed with all of this new data and new approaches, some colleagues set about analyzing it, and they were weighing up different potential scenarios for plant evolution, considering various different situations whereby different combinations of vascular or non-vascular plants might be related to each other in different ways, and asking which one the data best supported. And what the data supported was something that hadn't really been talked about for a little while. was a situation where the non-vascular plants, the hornworts, liverworts, and mosses, formed a monophyletic clade called the bryophytes, and this was in turn sister to the vascular plants. So all three of these clades are then more closely related to one another than they are to the vascular plants. We've kind of got rid of this successive branching situation. And since then, these next few slides are really just highlighting how since this paper, lots of different authors and studies have arrived at this same conclusion of monophyletic bryophytes, regardless of, for example, which species they might sample, or the different methods that they've used, or the volume of molecular sequence data involved, whether this data comes from the nuclear genome or the chloroplast genome, and so on. But the studies are consistently finding this result, that land plants consist of two sister clades, the non-vascular bryophytes and the vascular tracheophytes. So while this might seem like a relatively minor tweak of the topology or the relationships among land plants, remember how I was suggesting how important the particular topology before was for our view of early plant evolution. And it turns out that this small change, the two monophyletic lineages, vascular and non-vascular plants, can completely shift our view of plant evolution. So whereas before the nature of early plants was predominantly determined by these non-vascular plants having to kind of successively consider these different non-vascular lineages, now it seems that both of these groups, the vascular and the non-vascular plants, have a much more direct bearing on this reconstruction. So I think the easiest way to demonstrate this is to look at the evolution of a particular trait, and that trait is stomata. So stomata are these tiny valves found comprised of kind of two kind of kidney bean-shaped guard cells. And in vascular plants, you find them pretty much on every aerial surface, and they have a really highly conserved function. They regulate water loss, they allow the exchange of gases for photosynthesis, so they're really quite important. On the other hand, in the non-vascular plants, they're quite different. So they tend to be found, if they're present at all, just on the base of the capsule, so here on a moss. But often they've been lost, so they're entirely absent in liverworts, but many mosses and hornworts have also lost them. And this particular distribution, just found kind of in this specific place, suggests that they may be involved in drying out the sporangium to allow spores to be released. But if I'm completely honest, it's not really known what they're doing in bryophytes. So if we know the distribution of traits among living species, we can then kind of work backwards to predict kind of the likely state in the past. And this is a process that we call ancestral state reconstruction. So here, for example, if we have a distribution of blue and green among living plants, we can predict the probability of the common ancestors being blue or green all the way back to the earliest plant. So if we look at our kind of original phylogenetic hypothesis, we can trace the evolution of stomata. So they're absent entirely in algae and liverworts, so will have likely evolved after the divergence of liverworts. What's more, because they would have evolved within this kind of paraphyletic bryophyte grade, they probably would have functioned as they do in a bryophyte like a moss, only later evolving this kind of fundamental role in gas exchange that we see in vascular plants. But under our new hypothesis, things are a lot less clear. So for instance, stomata could have evolved in the earliest land plants and then later on been lost in liverworts, but they also could have evolved independently multiple times in lots of different lineages. And what would this then mean for the function of the earliest stomata? Would it have been as it is in non-vascular plants or in vascular plants? So plant evolution has become a lot more complex. So one way that we could resolve this could be to look at the fossil record. And indeed, stomata are preserved throughout the fossil record. So the plant epidermis and cuticle is relatively tough. And in fact, more or less, as soon as we start finding macroscopic remains of plants, in each case, they seem to possess stomata. And in fact, kind of in our best preserved early plant fossils from the Rhynie Chert, we find that not only are there stomata, but from the work of people like Diane Edwards, we know that there is also a variety of stomata found across different tissues with different morphologies and in different distributions. So not only are stomata present more or less as soon as we find land plants, but they're relatively diverse. So even without really knowing where these fossils fit within our phylogeny, we can reason that not long after plants evolved on land, stomata were present. And the other line of evidence that we can use, again, forgive a quite busy slide, is to look at the genome itself. So we know that in the case of stomata, we know how they develop, we know how they form, and we know which genes are responsible for the formation of stomata. In this case, 3 transcription factors called speechless, mute, and farmer, each working in a kind of tightly regulated sequence to control the successive stages of development from what's known as a stomatal mother cell onto this kind of 2-guard cell around a pore morphology. And that's not really too important. What is important is that we know the genes that are responsible for the presence or absence of stomata in plants. And just as we can do for morphology or traits, we can actually retrace the evolution of genes along a phylogeny, identifying processes such as gene duplication, gene loss, or gene transfer, and also whether genes themselves would have been present in ancestral genomes. And we did this for these key developmental genes across a series of papers. And these figures, again, very busily show it in different ways. But what we found was that equivalent genes or homologous genes to these key transcription factors would most likely have been present in the earliest land plant. And what's more, whenever we find that stomata are absent, such as in lineages like liverworts, these genes have been secondarily lost. So kind of armed with our new phylogeny, we're having to work a little bit harder to understand early plant evolution. We're having to draw on multiple lines of evidence from fossils to genomes. But in the case of stomata, we can confidently say that the earliest plants would have likely possessed them. And if the earliest plants had stomata, then they must have evolved before plants. And further, in these lineages, like I said, that are lacking stomata, such as liverworts, they must have secondarily lost stomata in a process that we call reductive evolution. So stomata are really just a single trait. And hopefully you'll appreciate that there are kind of so many different aspects of plant biology that we can now start to reconsider in this new framework. So from different aspects of plant architecture, such as the evolution of vasculature and branching, to different reproductive features and life history traits, such as spores and the alternation of generations. So looking forward, we're really excited about the prospect of starting to evaluate all of these different aspects of plant form to ask the big question about whether the earliest land plant, the common ancestor of all living plants, may have, as it was the case with stomata, have had more traits or have been more complex than we might have previously thought. So to kind of wrap up, hopefully the main messages of my talk, in this era of phylogenomics, the land plant and the origins of land plants within the algae, these phylogenetic relationships have changed. And as a result, what previously seemed like quite straightforward stories of progressive complexity have now become a lot more convoluted. From the common ancestor of plants, we kind of have these 2 independent lineages, the non-vascular bryophytes and the vascular tracheophytes, and each seems to have kind of evolved along a different trajectory. And that going forward, there's a whole world of different plant traits that we can now reconsider in the light of this new phylogeny. So with that, I've tried not to talk too much of my own work, but any that did sneak in there was done largely by other people, so lots of collaborators and generous funders. But Thank you all as well for giving up your evening to listen to me.
Thank you very much, James. That was a really great talk, and it brought in elements of the two previous talks as well, not least reference to the Rhynie and the first um, complete fossils of land plants. Um, can I just ask the first question, James? Given this sort of stomatophyte, you didn't mention that term, which is coined in one of the papers, um, earliest sort of land plant. What do we know about the genes that control stomatal development in the zygnematales? Have people looked for those genes in those plants? Because you're sort of inferring that stomata were sort of incipient in those first land plants. So can you say a bit more about that?
Yeah, it's a really good question and something I think about a lot, Simon. So the problem we have with stomata is more or less every plant fossil has them,
Mm,
and what we're kind of lacking is any kind of transitional form. So the genes themselves, those kind of key developmental genes, originate with land plants. So they're not present in those algal relatives. So we kind of have a window of evolution between algae and land plants where we know these genes are evolving. We know that stomata are forming. We just don't really have any clues as to how. So we don't have a kind of pre-stomatal fossil. So kind of everything happens at once, and it happens to be a time period where we just don't have the fossils.
mm. And is that because the environment was just so stressful? There were all these new innovation plants coming and going and evolving and going extinct at the same time.
I think so, and it seems from the genomes to be a really dynamic period of evolution. So when we look at when different genes or gene families evolve, the origin of land plant is associated with this kind of big bang of new genes. And they're all the genes that we know are kind of quite important for the development of plant form. So we don't really know how these new genes are appearing. Some of them we think are being horizontally transferred from other lineages. Some seem to be forming by gene duplication of existing ones, and others just seem to be appearing. Um,
Hmm. Okay, thank you, James. Now I'll ask some questions from the audience. The first here is from Nick, and he's saying, "Were fungi really essential for all land plants?"
um, that's also a good question. I'm not a great expert on this. There are, I believe, some lineages of land plants that don't form associations with fungi, but I think they are the exception rather than the rule. So again, kind of using this retracing philosophy of looking at how things are among living species, we can kind of predict that the earliest land plant would have had some association with fungi, and this I think plays out in the fossil record as well. So
Hmm.
certainly in the Rhynie chert, we see fungal associations more or less as soon as we start seeing land plants. Likewise, I think in genomes, all of the genes that we know helped plants to regulate these relationships with fungi seem to be evolving quite early. So I think a lot of evidence suggests that, again, as with stomata, more or less as soon as you have plants, you have these fungal relationships. So in some ways, it then kind of begs the question, well, when did that evolve? And I don't know that.
Hmm. Yeah, I mean, certainly from the previous talks we've seen evidence of mycorrhizas in the Rhynie and Aglaophyton and all that. So it's a really early occurrence, an association in the evolution of land plants. Now, a question from Amelia. In your opinion, where is the future of phylogenomics going? Yeah.
Yeah, yeah, so the one thing that I should caveat my whole talk with is that phylogenies and phylogenetic hypotheses are rarely stable. So while the origin of plants seems to be kind of ticking along quite nicely now, the field of animal evolution is more or less paralyzed because people can't agree on which was the first branch in animal evolution. So having said that, it basically means the debate is never quite settled. And we are now reaching a point where, the number of sequenced genomes is astronomical, more than we could have really imagined. So now that we kind of have all of this sequence data, the question is, why do we not just know how everything is related? And I think what we are uncovering is that this process of speciation that underlies uh, phylogenetics is much more complex. So, certainly in flowering plants, we're finding lots of examples of maybe ancient hybridizations, so kind of distantly related lineages coming back together to form viable offspring. And this is kind of wreaking havoc with the models we have. So I think
Mm,
phylogenetics needs to continue now that we have all this data, but considering kind of more and more complex evolutionary histories.
mm. And genome duplications are often associated with hybridization. So the instances of whole genome duplication that have been noted in the phylogenies of all sorts of organisms, are they now considered to be more likely linked with hybridization or just whole genome duplications in a particular lineage?
Yeah, I think maybe more the latter. We are getting better at untangling these kind of duplication and hybridization situations, but you've probably described the kind of nightmare scenario
Mm,
there, Simon, where we have to try and pick apart these kind of situations. We also know that, so maybe closely related lineages hybridizing and doubling isn't such a concern. But we also know from ferns that they don't necessarily have to be closely related. So very distantly related ferns seem to be able to produce viable hybrid offspring. And what that
mm.
means for kind of deep time phylogenetics is worrying, but
Yes, yes, indeed. It's quite a minefield. So we've got a question here. Again, quite a big question. Are there any big unsolved mysteries in plant genetics and evolution?
yes, yes, there are. I think there are lots. But I think one that I maybe alluded to a bit in my talk is that plants have a relatively unique life history. So as humans, we're kind of mostly diploid. We kind of have two copies of our genome in each cell. And for most plants, that's the same, apart from in the bryophytes. So these non-vascular plants spend a lot of their life cycle being haploid, so with just a single
Mm,
copy. And they seem to kind of alternate. So it's this alternation of generations between being diploid and haploid. And at some point, this kind of feature evolved. So plants started to mess around with these two generations and lengthen them both in these different lineages. And this is coming from algae that were predominantly haploid. So this is, I think, kind of one of the big questions in plant evolution is how these two
mm,
generations evolved and what the impact was for early plant evolution and how these two different generations seem to interplay, there's certainly a suggestion that there's a degree of conflict between the two. So I think that's a really big and exciting question.
Hmm. Thank you. Next question, and they're coming in at the moment. Are there any big changes in the chloroplast genome during the evolution of land plants?
Yes, I think so. And again, I'm not a great expert on this, but for a long time, the chloroplast genome was the kind of the backbone of phylogenetics because
Hmm.
unlike the nuclear genome, we didn't have these kind of crazy expansions and duplications, and the chloroplast genome is already quite streamlined, so we tend not to see big changes or the loss of genes. So kind of the photosynthetic machinery is very well conserved across plants. So I think there are changes in it. I think genes are gained, lost. We see inversions, so chunks of DNA kind of swapping around. But on the whole, I think the chloroplast genome is much more conserved than is the nuclear one. But yeah, not 100%.
And before this question, the poser Jazri said that was a fabulous talk. So, but the question is, any chance that there are genes in algae that have not yet been recognized as ancestors for stomata genes? And alternatively, is there any hint of gene transfer from some other organism being involved in the evolution or appearance of stomata?
Um, to first answer the first part of the question, kind of an algal precursor to stomatal genes. We know that they do have some genes that are involved in either stomatal development or function. So, a good example is that stomata open and close in response to water stress. And a lot of these water stress genes seem to be present, and the whole kind of stress response pathway is present in algae. However, kind of lacking stomata, these genes are obviously involved in some other process in algae. So quite a large part of plant evolution involves this process called co-option. So taking a gene that exists within the genome and is involved in one function and basically repurposing it for another. So that's quite a likely scenario for a lot of plant traits, was kind of taking genes that had some function in algae and then kind of finding them a new one in this kind of brave new world. To then talk about horizontal gene transfer and stomata, there's no record, in my knowledge, of horizontal gene transfer specifically related to stomata, but we do know that horizontal gene transfer has happened throughout kind of pivotal moments in plant evolution. So some studies have suggested that this kind of origin of land plants and terrestrialization was associated with a kind of higher rate of gene transfer from other lineages, typically kind of bacteria and fungi, than at other points during plant evolution. And that again might kind of tie into the previous question about a close relationship between plants and symbionts. But yeah, nothing specific to stomata.
Okay, now there's a really nice question coming up from an old friend of ours, Richard Bateman. Isn't the perceived monophyly of groups such as bryophytes, um, manilophytes (brackets ferns), gymnosperms, and angiosperms disastrous because we can no longer reconstruct the sequence in which characters were acquired by land plants, and he ends long live paraphily.
Um, I'll be honest, Richard, I'm not entirely sure I'd agree that it's disastrous. I would say that the kind of neatness of these kind of monophyletic lineages certainly ignores a huge amount of diversity in the fossil record. So when I talk about these kind of neatly packaged lineages, there were kind of lots of other interesting things existing at the time that maybe we will um, have forgotten. But
Okay, right. And then the final question, unless one comes in while I'm speaking or you're speaking, why does plant evolution seem to proceed much more slowly than animal? I'm not sure whether that is the case, but
yeah, I think I would agree with you, Simon, that it probably isn't slower. I think part of that may come down to our perceptions of maybe the degree of morphological change. So obviously, if we look at the transformation that occurs between a sponge and a primate, then that does seem like a gross remodeling of life. And to some extent, plants still kind of maintain maybe more recognizable features along their evolution. But actually, given that, what we think of as leaves in a moss and leaves in a flowering plant are actually completely different organs,
Mm,
I'd argue that plants have kind of undergone this same kind of fundamental remodeling as well.
mm. Right. Well, thank you very much, James, for that last answer. And thank you for a very thought-provoking talk this evening. And thank you all for attending. A big virtual round of applause to James from everyone. And we'll see you in 2 weeks' time for the next installment in this plant evolution series. So good night, everybody.
Mm.
Good
Thank you.
night. Thanks, James.